Hello, everyone. In this presentation, we will discuss common deficiencies in peptide sameness studies during ANDA review. Peptide generics are becoming increasingly important in modern therapeutics, but demonstrating sameness with the reference-listed drug can be challenging due to their structural complexity. In the next few minutes, we'll explore the most frequent issues regulators identify during review and practical strategies to avoid them. Peptide sameness is a critical regulatory requirement when developing generic peptide drugs. Unlike traditional small molecules, peptides are structurally more complex and may contain multiple conformations or sequence variants. Because of this complexity, regulators expect a comprehensive analytical demonstration of sameness. This typically involves multiple orthogonal analytical techniques to confirm structure, purity, and physicochemical properties. Ultimately, these studies ensure that the generic product will deliver the same safety and therapeutic effect as the reference product. During ANDA review, regulators frequently identify certain recurring deficiencies in peptide sameness studies. One common issue is incomplete characterization of the primary structure, where the amino acid sequence is not fully confirmed. Another major concern is insufficient impurity profiling, especially when peptide-related impurities are detected but not fully identified. Finally, many submissions show weak comparative analysis with the reference-listed drug, making it difficult to confirm true product similarity. The first major deficiency involves primary structure confirmation. Regulators expect clear evidence that the peptide sequence in the generic product is identical to the reference product. However, submissions sometimes provide limited peptide mapping data or incomplete MS-MS confirmation. Another common gap is the lack of verification of terminal residues, which can significantly influence peptide stability and activity. To avoid these issues, sponsors should ensure complete sequence coverage and robust analytical confirmation. Impurity characterization is another area where deficiencies often arise. Peptide synthesis can generate several variants such as truncated sequences, oxidation products, or demedated forms. Regulators expect these impurities to be identified and structurally characterized whenever possible. Problems occur when impurities are detected but not identified, or when the impurity profile is not adequately compared with the reference product. If unique impurities appear in the generic product, toxicological justification or qualification data may also be required. Direct comparison with the reference-listed drug, or RLD, is the cornerstone of peptide sameness evaluation. However, some submissions rely on analysis of only a single RLD batch, which does not capture the natural variability of the product. Another issue is performing analyses under different experimental conditions, which makes comparisons unreliable. Regulators generally expect evaluation of multiple batches of both the RLD and the generic product, analyzed using the same validated methods. Another common deficiency is the lack of orthogonal analytical methods. In some cases, sponsors rely heavily on a single technique, such as HPLC or mass spectrometry. While these methods are powerful, regulators prefer multiple independent techniques confirming the same structural properties. Additionally, higher-order structural features such as alpha helices or beta sheets may not be adequately evaluated. Techniques like circular dichroism, or NMR, can help strengthen the overall demonstration of structural similarity. Even when analytical methods are used, regulators may question whether those methods are appropriate and sufficiently validated. A common deficiency is lack of strong method validation, especially regarding sensitivity for detecting low-level impurities. Another issue is failing to explain why a particular method is suitable for distinguishing peptide variants. Clear validation data and scientific justification are essential to demonstrate that the analytical methods truly support the sameness assessment. Fortunately, many of these deficiencies can be avoided with careful planning. A strong peptide sameness strategy should include multiple orthogonal analytical techniques, ensuring that structural properties are confirmed from different perspectives. It is also important to analyze multiple batches of both the reference product and the test product to understand natural variability. Finally, sponsors should aim for complete sequence confirmation and thorough impurity identification before submitting the ANDA. To conclude, demonstrating peptide sameness requires comprehensive, multidimensional analytical evidence. Many regulatory deficiencies occur not because the peptide is different, but because the analytical characterization is incomplete or insufficiently justified. Using orthogonal methods, performing strong comparative analysis, and planning the analytical strategy early in development can significantly reduce review challenges. Ultimately, a well-designed peptide sameness study supports regulatory approval and ensures patient safety.